Hi, I'm Elizabeth Williamson, and this is my presentation about Drosophila and the SMN gene, taking a closer look at the tutor domain. So first, to introduce our subjects, this is Drosophila. So a lot of times in science, we do experiments on what we call model organisms because it would be unethical to perform them on humans. So this fruit fly species is often used as a model organism for humans because, believe it or not, we share a lot of similarities. For instance, their SMN gene is similar to ours. SMN stands for survival motor neuron, and that's what this gene codes for. You don't really need to know what it does, but in humans, we have two genes for this. And if both copies of our SMN1 are deleted, we will likely develop spinal muscular atrophy, or SMA. And so this disease obviously looks different between humans and Drosophila, but the gist is that it causes affected individuals to gradually lose their ability to move. So here is the life cycle of Drosophila. It's pretty quick, as you can see. It only takes about 10 days for an egg to become an adult. And the two main stages we're going to be focusing on are pupation, which is when a larva becomes a pupa, and eclosion, which is when a pupa becomes an adult. So here is the SMN gene. All of these arrows point to different mutations, which are basically just mess-ups in the gene. They're all located in different sections. So we have the GEMIN2 binding domain, the tutor domain, and the YG box. And you may think, why does it matter where the mutation is located? But now we bring temperature into the mix. So this graph shows the percent pupation, or the percentage of larvae that survive to become pupa. And the blue bars show the larvae that are raised at 18 degrees Celsius. The green shows 25 degrees Celsius, and the red shows 29 degrees Celsius. And each of these confusing-looking codes along the bottom of the graph represent a fly with a mutation in different parts of the gene. So as we can see, the ones with mutations in the tutor domain have barely any red, meaning that essentially none of the flies raised at 29 degrees, which is a higher temperature, survive to become pupa. We see red everywhere else on the graph. So that shows that raising the temperature doesn't have as dramatic of an impact on the survival of flies with mutations in other regions of the gene. And this graph showing the percent eclosion shows us some similar results. So again, percent eclosion just describes the number of pupa that survive to become adults. And we see even more clearly here that the blue bars for the mutations in the tutor domain are always higher than the green bars. We don't see that anywhere else on the graph, so that solidifies that these mutant flies need lower temperatures for maximum survival, even more so than the other mutants. So what can we take away from this? Drosophila with mutations in the tutor domain of the SMN gene are more sensitive to higher temperatures when it comes to viability. Viability is just another word for chances of survival, which we looked at with the percent pupation and percent eclosion. So this is only one part of the experiment's findings, and it was a very early finding, which is why I chose to present on it, because I think it's so interesting that the researchers weren't even expecting to find anything unique about the tutor domain, and yet it directed the rest of their research to focus primarily on what we call tutor mutants only, because these specific mutant flies have a lower percentage of individuals that survive to the pupal stage and a lower percentage of ones that survive to the adult stage. So that's the end of my presentation. Thank you so much for listening.